Hello everybody, welcome to our video this afternoon where we are going over biology chapter 2, reproduction. So, as always, let's get into it. So, this chapter has definitely a lot of important parts, but first we're going to start with mitosis. And mitosis is basically the process that all of our cells go through except for our sex cells, which we'll go over in a moment, but all the other cells in our body go through mitosis. And so what that means, let me stand on this side. Okay, so what that means is mitosis, right? We start with mitosis. You have prophase. So it's PMAT is a short way to remember it, but it starts with prophase. And in prophase, I'm hoping you can see the drawings. The DNA condenses, the centrioles migrate, so these are the centrioles here, migrate to opposite poles. The nuclear envelope disappears as well, and the spindle fibers begin to form. And here's what a picture of that. Here we're obviously simplifying, we're just using 2 chromosomes, but you know, we have 46 in total for as humans, so just, you know, making sure that we understand this is just a simplification. So we have next we have my metaphase. So PMAT, PMAT, prophase, metaphase. Metaphase is when the chromosomes align in the middle along along this plate in the middle. And yeah, so that one's pretty self-explanatory. Then you have anaphase. The sister chromatids are separated anaphase apart. The sister chromatids, that's important when we're talking about mitosis, the sister chromatids are being separated. These are sister chromatids. I don't have a marker handy, but we have sister chromatids is basically attached. The chromatids are attached by this part right here, which is a centromere, and then these 2 parts of the chromosome are sister chromatids. Okay, so those sister chromatids are pulled apart in anaphase of mitosis. Then we have telophase and cytokinesis, which is when the chromosome so sorry about that. When the chromosome decondenses. Set this back up here. Give me one moment. Sorry about that. Okay, there we go. Okay, when the chromosome decondenses and the nuclear membrane begins to form again, and cytokinesis occurs, which is the actual splitting of the 2 cells that you have now. So the result of mitosis is 2 identical daughter cells. Okay. And so, as I said, this process is done by all the cells in your body except for the sex cells, which we will look at in a moment. But the cell cycle is just to get a better picture of when this is really happening. So you have G1, S phase, G2, and PMAT. And so G1, S, and G2 are all part of interphase, and that's the majority of the cell's lifetime is spent in interphase. So we have G1 is you just have 1 chromosome here that's not replicated. It does not have a sister chromatid yet, so that's why I just drew this here. So in G1 the cell grows and there's just normal cell activity. Then you have S phase, which is where DNA replication occurs, and that finishes with a chromosome that looks like this, which means now there's sister chromatids. Now that you have sister chromatids, you can now enter mitosis and go through PMAT and do each of those stages we just said, and again result in 2 identical daughter cells. So this is how our cells replicate. Next we will talk about meiosis, and meiosis occurs only in our sex cells, so in eggs and sperm. And one thing to note before we get into this is that the sex chromosomes, as mentioned, so it's XX for female would be so XX chromosomes. If you have 2 X chromosomes, you're identified as biologically as a female, and if you have an XY chromosome, you're identified biologically as a male. And the Y chromosome in males have an SRY gene on their Y chromosome, which carries a lot of that, which is an important gene in the male reproduction, okay? And yeah, so meiosis, as we said, only occurs in sex cells. And let me go on this side here. So, meiosis. We have P1, so this is prophase 1. So you're going to see P1, P1, M1, A1, T1, P2, M2, A2, T2. And this is prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. So the result of meiosis, before I get into the exact steps, the result of meiosis is 4 haploid cells. Okay, and for mitosis, it was 2 identical daughter cells. Okay, but here we're creating 4, and they're not identical. And they're not identical because of the variation in the steps. So basically what happens for meiosis to occur is that for a gamete to form, right, to start having a baby, you have the chromosomes from the mom and you have the chromosomes from the dad, but they're both carrying a different, they both have chromosome 1, they both have chromosome 2, etc. But the actual genes on those, the genes on them might be different, right? Because people are different. We have different, even if we have, you know, a gene that codes for hair color, the mom might have one that codes for blonde hair, the dad might have one that codes for brown hair. So we have 2 different sets of these chromosomes coming together when you're having a baby. And so what happens is that the first part of this, the P1, M1, A1, and T1, that is when is the reductive division, because we're reducing the amount of chromosomes we have. Because if here we can't just go straight into producing daughter cells because they already have too many chromosomes. So if you had an egg cell that had a copy of everything from the mom and the dad, and you had a sperm cell with a copy of of everything, all the chromosomes from a mom and a dad, and you put them together, you have way too many chromosomes for humans. So what we need to do is have reduce the number of chromosomes that you're actually putting in these gametes, so that when they come together, they're able to form a full human. And with mitosis, our body cells don't, come together like that. They're just replicating. So they can go through mitosis, and they also do not have the homologous pairs that we see in meiosis. So the homologous pairs are, like I said, chromosome 1 from mom, chromosome 1 from dad. Those are homologous pairs. Chromosome 2 from mom, chromosome 2 from dad. Those 2 chromosomes are homologous pairs. So we see in meiosis, is the first half of meiosis is them reducing those. And what I mean by that is in prophase 1, you see the same steps here, okay? So DNA condenses, centrioles migrate, top set poles, and the nuclear envelope disappears. But you also see crossing over, and crossing over is an important step in prophase 1 because that gives a little bit more genetic variability. And yes, so you see this is chromosome 1 from mom, chromosome 1 from the dad, and let's say, this is a chromosome 2 from the dad and chromosome 2 from the mom. So now they are together and you are crossing over. So this gene up there might switch with this gene right here because they're so close together, and they might swap a gene or something like that, which creates genetic variability. So then we have metaphase 1, which is where the homologous pairs line up along the metaphase plate, and the spindle fibers attached to homologous pairs. Here we are attaching to sister chromatids. Here we are attaching to homologous pairs, at least in the first set of this, the reductive set of this. Okay, so in anaphase 1, you're pulling apart the homologous pairs, which results now in you have these 2 cells. So you do telophase 1 and cytokinesis 1, and now you have these 2 cells. right? And these two cells can now go through the rest of the steps of meiosis very similarly to the mitosis steps, because now we've reduced the number of chromosomes that we're working with, so that when we have, you know, an egg cell meeting with a sperm cell, we have the correct number of chromosomes. So the important part to remember here is that after telophase 1 and cytokinesis, now you have these two. So the rest of what I'm about to show happens 2 times. So I'm going to ignore this one just for now, but it occurs twice because you're going to end up with 4 haploid bodies. So just picture the same thing happening to this cell on its own. So if we're taking this cell, then we go through prophase 2. Again, nuclear, it stays, the nuclear, the nuclear envelope is not present. The DNA is still condensed. And you go to metaphase 2, which is where now you have the chromosomes lined up across the metaphase plate, and the sister chromatids can be separated. Now we have anaphase 2, which is where the sister chromatids are separated, and then you end with telophase 2 and cytokinesis. So now we have 1, 2, and then if this one was carried through all these steps, you'd have 3, 4. So now you have 4 haploid daughter cells, and they're not identical because of the recombination that can occur. So that is the major steps of meiosis. Now, one important thing to remember also is that non-disjunction occurs when the sister chromatids don't separate properly. So that would cause, uneven distribution of the chromosomes that we're supposed to have. You could have one maybe has 3, and one cell would only have 1 chromosome, like that. And so things like that can cause mutations, basically. But that specifically, non-disjunction occurs when they do not separate properly. Okay, so next we're going to talk about spermatogenesis pathway. And so this happens in males daily from puberty to death. So we have, this is just a simple chart to really recognize what's going on. I'm hoping you can see it. I might be able to move it closer. Okay, so this part right here, we have spermatogonia go through mitosis, so it's replicating. Then that turns into more spermatogonia, okay? So then when it's activated, it turns into a primary spermatocyte. That primary spermatocyte will go through meiosis 1, and now it becomes haploid. That means it has 1 copy, right? We reduced it, like here, we reduced this, so now we have them as haploids, and this is a secondary spermatocyte. And then it goes through meiosis 2, and you have a haploid still, but now you have a spermatid. Okay, so if we were looking at this for a sperm cell, this one of these at the end would be a spermatid. Okay. Now we have oogenesis, which occurs in women, and so you have prenatal. So before a woman is actually born, they produce the eggs that they're going to have for their lifetime. So prenatally, you have oogonia, and those go through mitosis, and you get about 200,000 oogonia. Okay, and those get activated and create primary oocytes. And once that happens, this stops for approximately 10 years until the woman gets to puberty. And then once she has puberty, the 5 to 6 primary oocytes go through meiosis 1. Okay, so this happens monthly, starting from puberty. So now we get meiosis 1 for the egg cells. Okay, now this is called a secondary oocyte, and you'll get a first polar body. Okay, and then let's say that this egg is fertilized. That means that if it's fertilized, will get a second oocyte that will then go through meiosis 2. So an egg doesn't go through meiosis 2 until it's fertilized. And then we have create an ovum and a second polar body. So those are important steps in this pathway, in each of these pathways for men and women. We also have something here about the male reproductive system. So to start off, we have the testes, which, like we said, was a primary sex organ. The ovaries and the testes are the two primary sex organs. So in the testes, you have something like this, is actually seminiferous tubule, and you can see these. The diagram isn't as important, it just helped me really understand, but you have Sertoli cells here, which secrete nutrients and androgen binding proteins. Then you have these things inside here, which are going to be the sperm. And the same thing here with spermatogonia. These will become sperm. And then you have these cells around this seminiferous tubule, which are interstitial cells, and these cells produce testosterone, and those cells are stimulated by LH. It's important to know like what stimulates and so on. And then we have FSH, another hormone that we'll talk about in another chapter of biology. Stimulates spermatogonia in the Sertoli cells. So FSH stimulates the Sertoli cells to secrete the nutrients, and it also stimulates the spermatogonia, and then the spermatogonia become the sperm, and then when you get LH, the LH stimulates the production of testosterone. And the sperm pathway, also short form, is known as 7-up. There's no N, but we have this pathway just as a mnemonic to remember the steps in this sperm pathway. So next we have, which begins in the seminiferous tubule and ends in the penis. U is the urethra, and V is the vas deferens. And like I said, there's no N, but yeah. And then we have the ejaculatory duct as well. Okay, so the cancer, this is the last part of this chapter. Cancer is actually a mutation of a DNA sequence, and this mutation begins to replicate out of control. And when that happens, now it will start migrating to surrounding tissues, which is how the cancer spreads. So that's just basic how cancer exists and spreads. But cancer-causing genes, just a couple of vocabulary items. So cancer-causing genes are oncogenes. Proto-oncogenes are normal genes that regulate a cell cycle. So if you get a mutated proto-oncogene, now you have an oncogene because now you can't regulate the cell cycle. So it's going to just continuously divide and replicate. And that's how you produce tumors. But then tumor suppression are proteins that code for a decrease in division. So it's important to keep those things in check as well to try to limit the growth of a tumor. But those are the general points of this chapter. As always, this is a summary of of the topics that you should know. We also have, I forgot to mention the menstrual cycle. It is part of this chapter, but I'm going to do it in the next video under chapter 3. So I know we did a little bit more of we did the male reproductive system, we did the sperm pathway, but we do have the menstrual cycle for women that we will get to in the next video. But that is the basics of everything, and as always, if you have any questions, please put them below, and we'll see you in the next video.